Nucleic acids are the last group of macromolecules that we'll cover. Nucleic acids are responsible for the storage, expression, and transmission of genetic information. So they really are the instructions for life and the way that the instructions are read by the cell. There's two types of nucleic acids, DNA or deoxyribonucleic acid, and this is the storer of genetic information. And so again, the kind of word that's often used is this instructions for life or the blueprint for life. RNA is a related molecule. It's very similar to DNA. However, its job is to read the DNA or interpret the DNA and help that DNA become a protein sequence. The process in which RNA does that is something we'll talk about in great detail at towards the middle of the semester. So nucleic acids are also polymers. The monomer of nucleic acids is the nucleotide. So the monomer is the nucleotide here. And so each nucleotide has three things in it. They have a phosphate group, a sugar, and the sugar is a little different depending on if it's DNA or RNA. RNA has the ribose sugar, think ribose for RNA, and DNA has the deoxyribose sugar, D for DNA. And then the third part is a ring structure that contains nitrogen, so oftentimes called the nitrogenous base for that reason. And this part's really important because this is the variable part. So these two parts are always the same in a nucleotide. This is the part that will make the nucleotide different. So we can look at some pictures here. This is an RNA nucleotide. So it has the ribose sugar, and this is what makes it a ribose sugar. That is going to be different from DNA. And then here's the phosphate. So this is part 1 of the nucleotide, part 2. Part 3 is the base. And again, this is the variable part. In this case, this is the uracil base. In DNA, which is a nucleotide we have down here, this is what makes it a DNA nucleotide. So notice that's chemically different from that one. So here's the phosphate, here's the deoxyribose sugar, and then here's the base that's variable. And so in this case, this is the cytosine base. And notice that it's a little bit different from the uracil base. The nucleotides will come together through dehydration reactions, and they are linked by phosphodiester bonds. So this is analogous or similar to a peptide bond in proteins that we talked about, or the glycosidic bond in carbohydrates. So in this image here, this is the phosphodiester bond. So it gets that name because the phosphate ends up being part of it. So there's a phosphodiester bond, here's another phosphodiester bond, here's another phosphodiester bond. So this is a DNA strand that includes 1, 2, 3, 4 nucleotides. So just like with protein, you have the part that's repetitive. So this is the part that's just repeating. Notice it's just sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So it's called the sugar-phosphate backbone for that reason, or just backbone for short. And then again, kind of like with proteins, we see the variable region. That's this part here. And remember, the variable regions in nucleotides are bases. And so this is the part that's going to make DNA sequences unique, as the base sequence. DNA takes on a very particular three-dimensional shape called the double helix. And notice that this is just true for DNA, so not for RNA. The double helix, it gets the double part because it's 2 strands. And it's a helix because it's a coil. So here's the 2 strands. They're going to be held together by hydrogen bonds. Between bases. So this is 1 strand, a second strand, Here's the hydrogen bonds between the bases of the two strands, and that will hold these together. And then these will coil around each other, and this is the representation of the coil. So here's the one strand, two strand, forming a coil. And that's the double helix. And again, we'll talk more about this in a later chapter. This is a comparison of DNA and RNA. So DNA stands for deoxyribonucleic acid. It's because it has the deoxyribose sugar. They have similar bases, so they both have adenine, guanine, and cytosine as the bases, but in DNA you have thymine base, and in RNA you're going to have a uracil base instead. So 3 of the bases are the same, 2 bases are 1 base is different. So they're both made of 4 bases. I didn't say this earlier, right? So in RNA you have A, G, C, and U. In DNA, you have A, G, C, and T. So again, the thymine and the uracil is what's different. DNA will form the double helix from 2 strands. RNA will remain single-stranded. There is only one function of DNA in your cells. It's to be the genetic material. There is multiple types of RNA in your cell. We'll focus a lot on mRNAs. But there are also rRNAs, there are snRNAs. You don't really need to worry about these at all right now. We'll get into these different types of RNA later on in this semester. So we'll talk a lot more about nucleic acids. This is just a brief introduction to them.